Hello, I am Nanjala Ruth. I'm a student at Pwani University, Kenya, and I will be giving a talk on evaluating the accuracy of genotype imputation in the major histocompatibility complex region in selected African populations. These are my supervisors. Genome-wide association studies traditionally use genotyping arrays to genotype large sets of individuals, and in this way determine which SNPs have been significantly overrepresented in the cases than in the control, and in this way determine association to the disease. They are, however, limited in that they can only measure tag of SNPs from the over 300 million SNPs that are available. An alternative will be to perform sequencing, but then it is quite expensive and technically challenging for large sample sizes, which is what is required for GWAS studies. The solution here would therefore be to perform genotype imputation, which is basically the process of estimating missing genotypes using a reference panel. We have some regions in the human genome that have high variability and may be difficult to impute. This includes the major histocompatibility complex region. The MHC region has been associated to autoimmune and infectious diseases, the innate and adaptive immune responses, and also the organ transplant and graft rejection. It is therefore important to impute SNPs in this region, as it will help to increase the number of available SNPs that can be used for future studies. The objective of this study is to therefore assess the accuracy of tools and reference panels that are used for MHC imputation in African populations, and also assess whether sample size and genotyping arrays have an effect on MHC imputation accuracy. This study is important because MHC imputation accuracy has not been tested yet in Africans, yet they have a high diversity and an extended linkage disequilibrium. The methods that we are going to use are as follows. We will obtain the study sets from the Gambian Genome Variation Project, test it against 4 softwares, impute2 and Minimac4 are general imputation tools, while SNP2HLA and HiBag are HLA-specific imputation tools. We'll also be looking at 4 reference panels, as you can see here. The 1,000 genome population, the 1,000 genome Gambian subpopulation, the 1,000 genome African subpopulation, and also the H3 Africa panel through their imputation service. The sample size will be between 50 and 300, and we will also be looking at the Omni array and the H3 Africa chip. This pipeline will be incorporated in an Nextflow workflow language and using Docker and Singularity containers. The outcome of this project will help inform future studies on the appropriate software and reference panel that can be used for imputation of the MHC region in African population. It will also help inform them on whether genotyping arrays and sample size has an effect on MHC imputation accuracy. Thank you.